Hello, everybody. It's me, Mahmed. I'm just kidding. I'm not Mahmed. For all of you who are expecting Mahmed to teach the class tonight, I am not. I am not that. I don't even know who Mahmed is. If you're wondering why my instant instinct was to go for like a Middle Eastern accent, because when I was growing up, all of my like competitive math tutors were all Middle Eastern. Except for Dr. Lee. Dr. Lee? Dr. Lee of AVID, now known as Ardent Academy. But hello, everyone. Welcome to this like interesting special session we're having tonight, where Kevin will do an open exam with everybody. Joseph, can you pull up the Joseph? I want you here to help me. Joseph.
Hello, it's me, Joseph.
Can you pull up the key for 2014, and then you can check me when I'm wrong?
Okay. You reference our handy dandy catalog of tests that we have never used.
Which I didn't realize existed until like a week ago. We're going to post this to our YouTube channel. For those who are wondering, we will also have to post on the Canvas then, for those who don't have access to YouTube. But hello, welcome. This is not what we usually do in Baology, but I figured this is like a good chance for people to actually get a sense of how classes feel when you know it's sort of a little bit more of a casual experience, which is what you get when you take our classes. So for anyone who's not familiar with us, we have a we run it's a tutoring company, but really we're more of a community. You know, feel free to come just to join us or to take our classes if you are interested in USABO stuff. But we're going to 2014 USABO. People wondering why 2014, Kevin? Well, this test actually holds particularly special significance to me, because 2014 USABO was the last USABO Open I took. Wait, actually, no, that's 2015.
So why are we taking this one?
Well, this is the second one, right? This is the second one that got me to the finals. Okay, we can do 2015. All right, do you want 2015? We'll do 2015. There we go. All right, we're doing the 2015 Open because this is the last one I took. That got
Kevin, we don't have the key for 2015.
Yes, we do. It's in the same folder.
It's not.
I kid you not, it is literally right here. It is one file down.
Oh, you're looking at the other folder. Oh. Okay.
Terrible. Because anyways, as you can see, our class is super great. But anyways, so we're gonna do this. It's the last time we did an exam. I took this exam and made finals. This test was probably a lot easier than what the OPAs look like today, but I think a lot of the ideas from this era still apply to how you take tests. And also, we can just see sort of where I am compared to where I was in that year. I think I got a 35 on this test because I low-key didn't like try. It was senior year, it didn't matter. Like, the cutoff was like a 27, 28. Cutoffs were very high at the time. They've actually gotten lower now. This test has gotten harder. It might be around 25, yeah. Cutoff around this time was generally higher than it is now. Today it's usually low to mid 20s back then. We actually at some point were theorizing that eventually the usable cutoff will be 40 plus for semis. That day never came to pass because, so this is interesting, I think a little history, like the meta shifted. Instead of the students getting better and better, which they did, the tests got harder and harder at a faster rate than the students got better. Well, people were getting better. The cutoffs were rising every year. We were afraid of a 30 plus cutoff. That seemed possible, right, at the time, because these tests are very easy, as you'll see. So let's do I'm going to look at the question. I haven't seen the answers in a long time, and we'll do it, and we'll see how it goes. And I'll tell you my logic thinking through it. Question 1. What best describes a nucleosome? That's easy. It's just B. All right, let's move on to question number 2. There's nothing you don't know what nucleosome is. If you don't know what nucleosome is, you can sort of maybe try to guess. The only thing I would say yosome means body. So I like A is actually a pretty reasonable one, but it's weird to call it the region because region wouldn't be body, right? C doesn't seem to be correct because Arnebaut doesn't seem to have anything to do with the nucleus. You can try to answer in the chat. I won't be reading chat till after I answer. A region in the nucleus where ribosomes are assembled. That's also, again, possible. It's like a body and nucleus. So if you don't know what it is, it becomes hard, but we obviously know what nucleosome is, and the answer is B. Number 2 is actually really interesting. This was a question that sort of pre-warned the introduction of multiple true false on the open exam. I don't think they do them anymore technically, but this was when they were introducing it at the time. You observe fly cells that are undergoing meiosis and treat them with a drug that inhibits the separation of parental homologs. Okay, you see it's parental homologs. That means there's two parents involved, and it has to be metaphase. If it's parental, it has to be metaphase. If it's homologs, it's metaphase. What it's one. So thus these cells suspend and metaphase 1. So 2 is also B, right?
Answer is AB.
Oh, so it would be sister chromosome metaphase 1. So it's homologous. Wait.
It's over.
I thought the separation of maternal and paternal chromosomes, but I guess yes. You see, this is what happens. I had misunderstood. Yeah. Okay. Am I crazy here? Who here thought parental homologs meant like the chromosomes from both parents? Okay, that's what I thought. That's why I said metaphase 1. But I guess here parental homologs is referring to the same homologs from one parent, which so you see, huge L, misread that. This does happen. In this case, it's tough, right? It could have gone either way. I totally thought it meant like from both parents. So I thought metaphase 1. It turned out this actually meant from one parent, so it's metaphase 2. But the logic still stands. A lot of drugs actually do this. The most famous one, of course, being colchicine. Nowadays, if you see this question today, probably they wouldn't even tell you this. You just say treated with colchicine potentially would be the question. Although that's more of a 20, I would say 16 through like 18, 19 sort of question where they love trivia, but now they're probably going to have something more similar to this, but there'll be like a figure from a paper that makes no sense before the question actually comes.
We're going to see that.
Not on this test.
Okay, but eventually, when you guys
if you guys take the booster classes, you guys will see that. And you guys will see Joseph and I mauled when we get to the place to get them wrong.
And you will mauled with us.
Yeah. Joseph and I actually pre-do the tests before those classes, so we actually know the answers going in. And then we mauled again, a second time.
Yeah.
Double number fun. 3. Psoriasis is a skin disorder that is caused by blah blah blah. What speed is really important in the USABO. So I would skip all this. I would skip at the beginning because it's not about psoriasis. But I see DNA promotes supercoiling. Which enzyme does this drug inhibit? This is a question where I would say the USABO giveth because they accidentally bolded the answer here. Notice how C is bolded. So the USABO giveth the answer to us. Topoisomerase does indeed unwind DNA and reduce supercoiling. The answer is indeed C. I hope. Maybe
we'll see more scenarios where the USABO taketh away.
The USABO, we always say the USABO giveth and the USABO taketh away. And this is one of the rare cases when the USABO giveth.
Yeah, so there's only like 5 of these total.
I'm gonna say this: even when you see something like this, you should still read the question because there have been multiple times when the USABO giveth, but it's actually fake and they actually take it away. It has happened multiple times. So I would actually still read the question. There, of course, are also times when the answer is obviously something, and then you just get it wrong because it was like, just kidding. Plants are animals, and then you get the question wrong. So that's pretty bad. Number 4, which of the cell cycle is treated with the drug questions they become arrested? So they can't finish. So it's E because it was supercoiling. Your cells cannot finish the replication, so that becomes suspended in S phase. Although here's where it gets complicated. If you know too much biology, you know cells cannot be arrested in S phase. S phase always goes to completion. So a more accurate answer is probably G2, but given that I have years of experience with this and I know how these tests work, this was before the meta advanced this far, I think the answer is E. But today the answer might actually be C because S phase always goes to completion. Joseph, is the answer E?
Answer is E.
See what I tell you. But technically, like even if this happens and your DNA shatters, S phase still finishes, right? It'll do like a really half-assed job and like only replicate the parts that it will replicate and then leave it out. This is 2015 USABO open, as you can see here. So as a result, you just have to sort of live with that. That is sort of just reality. Like not every question is gonna test the limits of your knowledge. If you saw this question on the open again, which you won't see because they don't have questions like this way anymore, I would today probably pick C, but E it's hard to say which it would actually be. So you had to use your intuition to try to figure out. You can try to figure out who wrote the question. There's only so many finalists. And they write most of the questions. So there's only so many, and you can always do that. It's a not invalid strategy. Almost every finalist I know currently would put C, except maybe Richard. But other than that, they would all put C. Some people might put E, but yeah. Yeah, so the idea is if you inhibit topoisomerase, the DNA supercoils too much and it breaks, or the tension becomes too high and replication stops. That's not actually what happens. The cell is very insistent on finishing replication. Like, you could like physically block the polymerases, they'll still try to do something. They'll find a way around it. They'll cut the DNA off so there's like no more DNA and finish. And they'll say S phase is complete because only a checkpoint exists in G2, right? The idea is like you could be smuggling drugs from like the US, like from LA to, I don't know, Vancouver. Okay, you're selling drugs. And if there's no police in between like San Francisco and Seattle, then there's no chance you get caught, no matter how much drugs you have. But like once you get past Seattle to like the border checkpoint with Vancouver, you do get stopped, right? And so like no matter what's happening, you will always manage to reach that checkpoint. Without a checkpoint, there's no stopping you. There's no arrests. See how clever I am? I'm so smart.
Genius.
Yeah, truly a genius. Even if you start like using your drugs on the way between San Francisco and Seattle, there's no police. It doesn't matter. You still won't get caught. You still won't get stopped, right? Don't do drugs. Drugs are very bad.
Drugs extremely.
Yes, I'm actually extremely anti-drug. I'm very chill, but I think drugs are very bad. They have a lot of mitosis.
Yeah, contrary to what you would expect.
Yeah, there are a lot of mitosis. What do you mean contrary to what you might expect? Anyways, number 5. A liquid culture of yeast was grown under anaerobic conditions, but then had a significant quantity of alcohol dehydrogenase inhibitor added to the medium. Which of the following would best describe the effect on glycolysis due to the presence of the inhibitor? I didn't read the question. I read it out loud. I didn't read the question at all. So if you don't have alcohol dehydrogenase, you can't make alcohol. Which is only important if you're doing anaerobic, so thus glycolysis would basically not happen, so it's E. Very straightforward question, right? You need fermentation to recycle the electrons from NAD to turn NADH back into NAD+. So that's what you make alcohol for. In yeast is actually so in humans, lactic acid is just a molecule that we can recycle back into pyruvate for some energy, so you still get stuff out of it. In yeast, the alcohol actually plays a purpose. It kills your neighbors. It's truly an Alex Jones type moment where you basically kill your neighbors, they die, turning them into food, and then it also means that whatever oxygen is left, you'll have more access to because your neighbors are dead. So it is like it's doubly good for the yeast to do that, as opposed to for us, where the lactic acid doesn't really do a ton. It's just sort of a recyclable product. Note that yeast cannot actually metabolize alcohol in most cases, but like in terms of breaking it down for their own use, but some can. The C-make is specified by the DNA using hydrophobic interaction. Okay, it's a little odd. By finding specific C-make and encourage television, or the Hall television, one machine can one make the C-make. So we clearly read hydrophobic. So thus you have to change something hydrophobic to something not hydrophobic. So in the first one, we change alanine to T, which is not tyrosine, threonine. Threonine is hydrophilic, alanine is not, so A is possible, right? B, you see the change, we change this glycine into another threonine. Okay, this is interesting. And then oh wait, is this DNA? Oh
yeah.
The first two are wrong. Unless you memorize, sorry, I did not realize. I was like, these two could both be right. No, wait. The DNA ones aren't right. There's no way they expect you to actually memorize the codons. You can if you really are like a mega nerd, but there's really no point. So this one, GVVLIA, this is everything in this is hydrophobic. Notice. So we're looking for the most hydrophilic. So so alanine, phenylalanine, phenylalanine, leucine, proline. Okay. Phenylalanine 1, so 2, and then this one is T. Okay, it's E. See, these are, yeah. I also thought these were amino acids when who was
they could have done all of them as just like G, C, and like T.
That they could have really made it so you had to read the first part, but I always skipped the. I couldn't. So Joseph and I have what's called USABO vision, where we literally just don't read the stuff that doesn't matter.
And like it works like a
It-
good amount of the time. It's like
It
crazy.
works really well. It's really important for semis with the time crunch is actually like a problem. VLL, what do you mean? What's VLLFP? So the latter three are amino acids, the first two are nucleotides. No, read the answer choices. Reading is very important. Yeah, reading is mega important and usable.
Reading S tier skill. Yes.
Yeah, mega S tier skill. If you guys want, we'll release the usable skills tier list.
Yeah. Number one will be reading.
Number one is reading. Reading is so helpful. Replication inhibitor in the middle of what phase of cell get would these cells remain at? So you add it to stem cells in the middle of replication. I think this question, they're actually trying to say that the actual answer is E here in this question 7. I think what happened is the person wrote 2 versions of the same question and they used both because they were short on questions. So I think the answer to this one is actually E. Am I right?
Answer is D.
Oh my god. Okay. Well, I mean, hey, you win some, you lose some. There's no way to know. Remember, this is basically the same question as 4. Like, I probably should remember that it is 2015. This was before people realized in the usable like general consciousness that E is true. One thing to remember is that usable biology is different from real biology. Like, there's this collective consciousness that we all share. So as well, some information just is gained or lost over time. And at this time, this was definitely not a well-known thing. This was known, but it wasn't something people were thinking about. So that's how usable develops, right? The consciousness, you guys are not fighting against other students. You guys are, in terms of the test difficulty, you guys are competing about, AP bios are also very different from bio. You're competing against the like usable collective consciousness, which has had like decades to grow, right? Like every person that passes through it, like leaves their like, thoughts scattered inside the usable consciousness. And that's a very interesting thing. Okay, number 8. The structure known here is AZT based on the chemical structure, how it'll work. I literally just read AZT HIV in the last sentence. That's all I read. So clearly it looks like a base. So the answer is clearly this is really dumb. I actually know exactly what this does. It mimics cytosine and it gets inserted and then it doesn't really work. But I don't know if they're looking for A, C, or E as the answer because all 3 happen.
This is just one of those moments.
This is one of the moments that I think I know a little too much, right? And that's the issue. I'm pretty sure they're not going for E because you can't intuit E from the thing without actually knowing it. I'm gonna go with C as the answer, Joseph.
Answer is A.
Okay, yeah. So what this does is this mimics cytosine gets inserted. What it does is it basically creates like a transcript that doesn't insert properly and gets degraded. The issue is it only works on HIV and not on humans because reverse transcriptase has lower fidelity than the human DNA polymerase. Now, I can see now why C is bad because it says it implies that this molecule decreases the fidelity of reverse transcriptase. So if I maybe spend a little more time thinking, I probably think A would probably be a better answer. E is also true, by the way, because random DNA hanging out does get deleted more because this version can't insert as quickly or as well. So A is a better answer. And so this is an L on my part. Clearly, I wouldn't have made finals if I've taken this. You have to be really sharp. Like I'm not in competition like when I'm in like competition mode, like when Joseph and I like grind all the opens for boosters or all the semis for the semis booster, we get into the mindset. We get like early on, it's so hard because you're like this question is so dumb.
Yeah.
But as we go later on, your mind actually becomes very sharp and become pretty good.
Yeah. But also it sort of degrades because we end up going to goon mode.
It does. The goon mode is very severely bad.
Goon
yeah,
mode, it's an active fight between like getting into the usable mode and like fighting goon mode.
Yeah, goon mode is so powerful. No, but this drug is actually really interesting. It was one of the first anti-HIV drugs specifically. Very cleverly designed by the biochemist who made it. But yeah, so for anyone who this is the kind of logic you need, right? You need to be able to read quickly, think quickly, know stuff quick, recall quickly. Good for you, TSR. Everyone, let's get a good for you, TSR in the chat.
Good for you, TSR. Good for you, TSR. Double thumbs up.
Yeah. This is would be a good time to have Ann Wu, who would hit you with the Mars rover, looking for who as me. Okay. Number 9, you've been asked to identify a bacterial culture using a light microscope and examine a single bacterium, oil immersion, about your techniques. A is wrong. I don't even know what this means. B is wrong. You can still touch the lens if you wanted. C is kind of correct. D is correct, and then if fashion the cover slip, E is correct. So it's between A and B. Remember the not over here. I really don't think A happens. Like maybe B is true. So I'm gonna go with A. Joseph?
Answer here is B.
What is a numerical aperture?
It's like the aperture number.
Oh. I remember. I remember. Okay, look, I got it out of 2. It's pretty good. Clearly, I'm not as good as I used to be, but numerical aperture is the how open the lens technically is. What happens is they mean numerical because the actual aperture doesn't change. When you reduce the amount of light you lose, the technical aperture is bigger, so you're getting more light technically. That's why it's okay. I forgot what the aperture was. That's my problem. So the answer is B. Yeah, I was like, B seems dumb. I'm like, maybe you can feel the oil when you touch it, and that was clearly an error.
Your first mistake.
My first mistake was assuming that the test. Look, this one's obviously on me as well, right? But like getting it down to 2 is pretty good. Oil immersion again lets you do all the things here. This is actually a pretty good question about oil immersion. So yeah, B is the answer. Number 10, which is a lot of questions about mitosis and meiosis on this one. If you read those 2 chapters, you got like 6 free points. Which of the following is false about meiosis 2? Cytoplasmic division occurs. That's true. Individual chromosomes line up. That I think B is false. Sister chromatids separate and move to opposite poles. Okay, C is true. D is false. D is falser. Answer is D.
Answer is in fact D.
See how good I am at this? When you're doing this, it's really important. B and C don't contradict each other. 2 sister chromatids make up a single chromosome. Here, if it gets a little bit weird if you like think about as like a chromosome as each sister chromosome as individual chromosomes, which is why I was like B seems kind of wrong. But then when I read D, I'm like, these are so much more wrong, right? Like people will think of 2 sister chromosomes as 1 chromosome. That's perfectly fine. But it's depending on how you're thinking about it. But yeah, with questions like this, I usually like, you guys don't have this luxury. But I, on paper, when back in the ancient times, we had this thing, it's called paper. It's very similar to an iPad, but instead of the screen changing, you just had to get a new sheet of paper with different things on it. It's very similar, but you can write on it just like you write on an iPad. It was interesting, but I would mark physically like I would like cross out the answer if I didn't think it was possible. I'd like put a dash next to it. I would put like a dot or a dash next to it or a question mark if I wasn't sure. I put a question mark. If I thought this could be the answer, I put a dot or like an arrow or something to indicate I was thinking about that. So yeah, that's how I used to indicate all of these things. I know that like one of my students, Ian, bought a stack of transparencies and he put it on top of his thing. He would mark the question and just move the transparencies.
That is a little cracked, I will say.
I mean, it's smart. It's smart. I mean,
I wouldn't have thought of that.
I saw no issue with that. So you could do that. Yeah. And you can keep the transparencies.
Yeah.
Any notes you wanted to take on the question could be kept on it. I was fine with that. Like, it's basically scratch paper, which is allowed. Okay. Number 11. Spores and seeds in the site of reproduction, showing false. Spores contain communophytic tissues. That's true. Seeds contain both communophytic and sporophytic tissues. A seems questionable. Spores produce mature communophytes. Seeds produce new sporophytes. B is true. C is true. D is what the D is very false. And then E is True. So I would say it's D.
Answer is D.
A is true though. The seed coat integument is sporophyte tissue. I was saying A is like maybe sus depending on like how you're thinking about it. But yeah, D is just way falser. This question you can also sort of cheat. You can notice that they for some like that this statement appears twice, which means that the answer almost certainly is one of these two, and then these two statements are like just opposites of each are like pretty opposite of each other. So thus, like it has to be A or D, right? Like it comes down to that. Especially at this time, tests today are maybe a little better, a little more creative, but like tests at this time had a huge issue with repeating answer choices or like very similar ones. So that's something that you may want to consider. Okay.
Yeah.
Number 12, if one were to compare the disbursement of spores and seeds, the following would be false. A is false, but okay, we'll just well, no, A is not false actually. It says seed plants. I didn't know it says seed plants. B, non-seed bearing plants are a parabolic group. B is true. Seed plants produce spores developed in sporophytes that are dispersed. Well, I guess the pollen are dispersed, so I guess C is true. That's the issue with A is that A is kind of false because like pollen sometimes is like just anyways. D, non-seed bearing plants disperse spores by wind, water, active discharge. Where does the parentheses end here? Is this the regular all the rest of this?
The rest of the test in the parentheses.
Okay. D is true. Seed plants disperse seeds via animals, particularly insects. I mean, E does happen. It's not often, but I'm pretty sure E is false. Joseph, is it E?
The answer here is C.
I knew it. E was not gonna happen. Seed plants produce spores developed in the sporophytes that are dispersed. This is objectively true.
USABO, take it away.
I mean, this is objectively true, right? You all read this? Like pollen exists. Like pollen is a sporophyte. Oh, wait a minute. Pollen's a gametophyte. But pollen is also made of spore. Yeah, okay. C is wrong. I realize that spores develop into gametophytes. So gametophytes is dispersed. That's the issue with this question. So this is one of those tests usable. Everything is right except one tiny detail, and the spores develop into gametophytes, and I missed that. Yeah, I was thinking pollen coat, but yeah. I was thinking that spores can count as like some people can count pollen as spores because sometimes they undergo meiosis after the release, and it's very complicated. I would not personally get super duper caught up in this. C's, but I mean, E is wildly also incorrect. I mean, like you don't disperse seeds via insects. Like I can think of like a few examples, like some like beetles will like bury caches of seeds and that can help, but yeah, like it's not particularly. So E's also wrong, but USABO take it away in this case. I read E, I'm like E's so wrong, right? E's like, but DC has a much more structurally like unsound issue with it, which is spores develop into sporophytes. That's not true. Yeah, but there's like a few cases now. Particularly insects is still really weird, but whatever. Number 13, monoclonal dicots are angiosperms. Great. 20,000 species, 50,000 species, well, true for dicots, right? See, this entire sentence completely useless. You get in the habit. I said over the true for dicots
backwards.
Yeah, we begin with dicots. Okay, are a paraphyletic group. Dicots are in fact, they are a paraphyletic group, because monocots are derived from dicots. So including only dicots, not monocots, makes them paraphyletic. Have ring-shaped vascular bundle, but no pith in the stem. So remember that cortex in the middle, pith is on the outside. I don't think. I think B I don't think B is true. Have stomata in their roots. That seems false. Have the order of plant components from the medial, so that's the center, to the lateral at the side in the phloem. No, it goes xylem phloem endodermis. Includes the orchidaceae is actually so rosaceae is roses, orchidaceae is orchid, that's what she is. Asters, orchidaceae is monocots. So Do I just eliminate all of them? The answer is B. Oh, so the answer is A. Yeah, you're right. I said the answer was A. My apologies. I got confused.
Okay.
Yeah. Answer is A. I'm like, this is not. I don't like reading me. I'm like, this is not right. Yeah, it's A. Like, I confused myself. Yeah, answer is just A. Okay, great. 14. A lot of plant questions on this quest too. Angiosperms are dominant in most terrestrial ecosystems. Good for you. Which of the following does not contribute to dominance? Blah blah. Cuticle compared to ferns. Ferns have a cuticle. B. Stomata compared to liverworts. That's true. Like, lignin compared to mosses. This is also true. Seed compared to ferns. Well, ferns don't have seeds. So right now we're at A and D as possibilities. And then E, xylem compared to conifers. Conifers have xylem. So the answer is either D or A. Now, here's where it gets a little weird. This says dominance over specific terrestrial organisms. Well, like ferns obviously don't have seeds, so we know. So wait, it's which is one does not contribute. So the answer is going to be E. Great, easy. See, notice how you can still catch yourself even when you misread. Joseph, does the answer key say E? I have a feeling that this one might not. I have a feeling. Joseph, I can't hear you. Your audio's gone. So you were talking, but I can't hear you.
Can you hear me now?
Yeah, I can hear you now.
Okay. Yeah. Answer key says A.
Of course it does. I knew it. I knew it. The moment I read it, I'm like, The moment I read it, I knew that the answer key was going to be wrong on this one. So you could maybe say it's E because the cuticle in ferns and antifers are very similar, but like the xylem in conifers is different from the xylem in angiosperms in that they don't have in that they only have tracheates and don't have vessel elements. The thing is, like, it's really unclear what vessel elements, how they're better, really, and it's anyway. If you picked E, you got scammed here. I mean, the answer should be E, but I was like, I felt fussed about A, and I'm like, reading the question, I just know. I felt it the instant I read it. I'm like, USABO's gonna F with us. You know why? Because they had ferns twice. And during this period, if they have ferns twice, the answer is gonna have to do with ferns. There's no way they did it. Conifers do have xylem, yes. I just knew, Joseph. Are you not impressed by my intuition when it comes to this question?
Intuition, very correct.
The moment I read it, I'm like, I just know the answer is going to be one of the two. But I was like, xylem is a more principled answer here. Like, arguably, vessel elements are less hard to clog. They have less issues with air bubbles. The other issue with conifers specifically is really weird to me because conifers actually cover more land area than angiosperms, like as a whole, technically, right? Because there's more boreal forest and there's the citrus forest. Maybe not more than citrus forest, but it is the largest biome. Anyway, it's very confusing. And vessel elements are faster, less likely to get clogged, but that definitely matters for like say trees. But like head to head, conifers like ash trees are the same. The reason why angiosperms are so successful, at least in my opinion, is grass. Grass just blankets everything and like it edges out competitors. So it ends up being a very like dominant force for partially for that reason, right? Like grass is very powerful, but grass does not really need vessel elements. I don't think they really benefit from it. There's a lot of the earth biomes are grasslands because grass takes very little water to grow, and like they edge out other things. So that's why. Anyways, look, this is what happens. USABO will take it away sometimes.
USABO does take it away.
Number 15, which of the following statements about long distance transfer system in plants are true? A is false. B is true. I mean, B is true, but I'm not sure Visible Meta at this time knew this. C is true. D is true, and E is false. No, so I'm torn between B and D. Driving force that moves water in xylem over the end of the sun, that's true because transpiration, right? But B is also true. See, so okay, so if we were to use the rule from before, they mentioned phloem 3 times, xylem only twice. So that's the answer should be one of the phloem. So going with that, I'm gonna go with B. Both B and D are technically true. But I'm gonna go with B here.
The answer key says B, C, and D.
Oh, it was C is also true.
Okay.
Okay. This was a select all. No, oh, this was in the era when select all didn't have to tell you. Notice that are true. So we all missed out on this one. None of us noticed. It's B, C, and D. Well, what can I say? This was the era when they wouldn't tell you this. You had to know that I come from a very different era, like back when this was like really huge and like a really big problem on the test. Like it was actually like really bad for the meta because there's also all or nothing grading at the time. Which we're doing again now. Okay, so number 16, you are given an Arabidopsis mutant pin 3, which efflux protein disabled, which is the following, which you expect. So without the efflux protein, you don't have polar transport of auxin. So okay, but the question only asks basically what auxin does, right? Auxin doesn't deal with germination, but it does have to do with phototropic response, and nothing to do with fruit ripening, nothing to do with stomata. That's so this, yeah. So this is ethylene, citric acid, and these two are like citric acid and gibberellins. That's for C.
Answer is C.
Yes.
Very good, very good.
Okay. Number 17, Chrissy is a papillon. When I read this question, I thought papillons were insects. I know what a papillon was. It's a dog, it turns out. And this is actually Mrs. Frame's dog. Number 17, Chrissy is a papillon, an herbivore who tends there's crunchies with slash palms and grasses on the yard. Try to find a new different Bacchia. Different Bacchia actually has oxalate crystals. This is a specific diagram in Campbell's. And this is, I think, a tissue level defense. But the only way to remember this is to have memorized that one diagram in Campbell's. It's one of those all black pages, and it talks about different Bacchia, and I believe it's a tissue level defense. So the answer is D. Joseph?
Answer.
It's either, yeah, I think it's D, but it's A, B, or D, if I'm not mistaken, but I could be completely wrong.
Would you like to narrow it down?
Sure, I'll narrow it down. So it's caused by oxalate crystals. They're located in the cells. But it's only found in certain tissues. So I'm gonna go with D.
Answer is B. Okay,
well, the cards are found. Anyways, I don't remember the diagram perfectly well. It was only in one edition of Campbell's too.
That's fair. That's fair.
You know, it's tough. I knew this question when I read this. I knew the question because I seen the diagram recently. It was actually really broken because this was in an at the time unreleased version of Campbell's, and only the campers had it. So this was actually mega broken on release. This question. This is like when you like play
Campbell's, or
I think actually well, I haven't, I haven't read 12 cover to cover the way I did with 10 and like not 7. I can check. Like, cause I don't think they have them anymore.
Yeah, I don't recall reading this, but
yeah, they have these all black 2-pager sections which they would talk about things like this. Yeah, it's gone. They don't have it anymore. From
is it because like it was like too like inconsequential?
It was not good. These no, they still have some version of some of them, but I'm saying they still have different Bacchia one. I don't quite remember what chapter it's in. This is one of those like very mild like version difference type things that does sometimes come up in Campbell's like issues.
Your Campbell's is not up to date.
It's very
the latest version.
It is. In 12th edition, it is figure 39.27. They still have it, and it's the it's so it's the rayfied crystals in the taro plant as they use the example now, but back when I had it, it was different Bacchia. Yeah, and it was oxalate crystals instead. So anyhow, that's something you just have to know. 39 is plant hormone. That is plant defenses. It's weird how they break stuff down. Anyways,
Yeah.
look, I don't think they don't really do questions like this anymore. In part, I will say that in part because of the advancing meta, Campbell's is no longer as emphasized as it used to be. Like back in the day, like not knowing a diagram in Campbell's was really your fault. It was like just a failure of you to study enough. I don't think that's the case anymore. Right? Today, if you don't really study Campbell's enough, so you know like this specific diagram the way Joseph and I had to back in our day, it's not that big a deal. Like I think you'll get over it. But yeah. So this is one of the things that just happened. Oh my god, we're nearing the end. I guess maybe we'll hit question 20 and stop and be like, you should pay to see the full version, but there's number 18, in which of the following habit would you find many C4 plants? This question is a classic. It's not truly a certified hood classic, as some questions are. I don't think this test had very many certified hood classics, but C4, right? C4 does best under arid, like hot conditions. So subtropical grassland and tropical rivers canopy, I would say B. Joseph.
Answer is B.
Yeah. So arid and hot. That's the thing. So hot but wet is canopy better. Just remember that like corn grows better in like Kansas than in Hawaii, and pineapple grow better in Hawaii than in Kansas. Okay. Canopy is right because you're at the very top, right? There's not as much more, there's a lot of sun. Yeah, hot and wet is better for canopy. I think bromeliads, like humid. Anyways. 19, hypothesize that caloric intake may increase lifespan of C. elegans has a hypothesis. Which of the following examples of positive control for support your results? Knock out. Okay, so you want a positive. The positive control means that proves your thing positive. Like this is how you increase their lifespan, right? Knock out a gene that decreases. Knocking out the gene wouldn't do anything for diet because you have to do a gene that actually increases. Do something that increases lifespan, right? B. Provide one group of worms with a drug in order to increase the lifespan. B would probably work. This seems to be multi-select, so I would say B. C, provide one group with a normal diet measured with control. That's not a positive control. Provide one group with sugar water. That's a negative control. And then E, isolate one group of worms on a long enough genetics and provide them with the treatment diet. Ooh, E is tough because if they don't have a longer lifespan, that works. But yeah, I think B and E is my answer. Joseph?
That's A, B, and E.
A,
So
B.
A is.
A is not a positive control.
That sounds like a. I guess maybe it's
a color control for like eating.
maybe you're positively controlling for like the decreased ability to eat, the decrease.
No, they're eating more, right? Well, I don't know what this is positive controlling for.
I think like the positive result is like the decrease in ability to eat. Like you just want to observe that, but that's sort of weird.
You knock out the gene decreases the one's ability to eat, so that's you're increasing the one's ability to eat. So thus,
oh, wait, you knock the gene decreases the
you knock out a gene that decreases. Right, so the worms eat more. I don't know. I don't really think A is a positive control. It
doesn't seem like a regular kind of positive control either. So
yeah,
I don't know.
I was like, maybe it's for like, but I assume you're varying. Maybe it's a control against like how much the worm wants to eat. But that's more of a negative. That's still weird. Positive controls are controls that should give you the positive result, basically. Right.
I guess so. Like positive was like you want like a result, you see it, and like it's like it's there.
I just A would be like is A a positive? A is some kind of control. It's like a control against this one gene. I would say it's more of just like a test control rather than a positive or negative control.
That's what I'm thinking too. You can like
Yeah.
kind of make the argument for like positive control, but it's not good.
Yeah, it's
Like I guess for that reason, you can also say like C is also positive
yeah, that's my
control.
point, right?
Yeah,
Like,
that's not really.
okay, I don't know. A does not seem to be reducing it to me.
Yeah, A is kind of bad, but you know.
Okay, we'll do one more question.
Yeah.
Well, this one seems spicy because of, look at this. USABO giveth,
The youth giveth.
but you should still read the question. If you're running out of time, I would just pick E, but like it's very, very risky because they do mess up a lot.
I think they've only messed up like once where they actually.
No. It's like 3 times. If you count the semis, it's more than once. I think it might just be once.
Oh.
To do this, you simulate basically just looking at big versus small axons. So look at how I read the question in like 1 second. That's how you should be reading these questions. The giant squid axons are myelinated. We know giant squid axons are not myelinated. But based on what the following is true regarding the properties of your calculations. The issue, I'm not sure if the meaning like could theoretically be true or is actually true, but one is false. Squid axons are not myelinated. Only vertebrates have myelination. Culture mouse has myelinated, therefore speed is greater because there are voltage gated ion channels within the myelination. So 2 is true. C, the script is larger diameter. 3 is also true. And 4, the culture therefore is cellular population exponential. Oh, never mind. 2 is false. It's actually 3 and 4. The answer is E. So great. The USABO do give it.
USABO did in fact give it on this question.
Yeah, it's so 2 is wrong because you don't have throughout the myelination. I didn't read this last part. I just read greater because, and I'm like whatever, and I moved on. But 4 is more correct. So yeah, 4. USABO do give it here. The answer is E. All right. I hope this was helpful in sort of seeing how we logic through these questions and how we solve them. If you want more like of stuff like this, come join us on our booster classes where this is pretty much all we do, along with some lecture, along with like content review. And so yeah. All right. I hope this was helpful, more helpful than Roy's office hours. And yeah. However,
Thanks everyone.